Welcome back to Good Law, Bad Law. My guest on today's episode is Professor Henry Greely of Stanford Law School, where he's also the director for the Center for Law and the Biosciences. He's also the author of two books. One is just about to come out. First book, The End of Sex and the Future of Human Reproduction, and his just coming out book, CRISPR People, Science and Ethics of Editing Humans. So I think you know where this is going. We're talking about the development, the meteoric development of gene editing technology, what this means for the curing of diseases in humans, but also the big ethical questions about how this may be applied in editing the genes of future children and future babies. As well as all the different applications this could have on everything from agriculture to climate change to so many different ways that we hardly can imagine it. But Professor Greely can imagine it, and we start to scratch the surface of this topic in today's episode. I know my brain has been incredibly expanded just thinking about this and talking to Professor Greely. I know you'll enjoy this episode of Good Law, Bad Law. Stay tuned. So I'd like to welcome my guest on today's episode, Professor Henry Hank Greely, professor of law at Stanford, my old stomping grounds in the Bay Area of California. He's a professor specializing in bioethics and biotech, health law and policies, the director of the Center for Law and the Biosciences, and has really spent his career leading to this moment, not only talking to me, of course, but on the cusp of his new book coming out, CRISPR People, Science and the Ethics of Editing Humans. So first of all, Professor Greely, thank you so much for being on the program today. I've really been looking forward to getting into this topic with and really looking forward to speaking to you in particular. So first of all, welcome and thank you.
Thank you for giving me the chance.
I was you and I were chatting just before we started about kind of what the scope of our conversation ought to be today, and I was explaining that I have had a growing interest in the subject of CRISPR, the bioethical, medical, scientific, ethical issues raised by CRISPR technology, and then here you have a book coming out that gets at some of the really deep, both exciting, earth-shattering, but also in some cases disturbing questions that we as a species face when we think about the use of gene editing and human reproduction. But before we get to that, I think, and to help people understand what we're talking about here so far, I thought we could start with the subject of the coronavirus vaccine, because we are all so excited about the incredible advances that bring these new vaccines to us and can't get to us soon enough. But I think very few of us have actually thought about how these vaccines actually were created, the technology that's allowed them to be created. And so maybe we could start with that and have that lead into some of the ethical issues and bioethical questions that CRISPR technology raises. So is that something you can help us understand, both what is CRISPR, how that represents such a sea change in the development of new technologies like these vaccines, and particularly how they work in the context of these vaccines?
Sure, but as I'm sure you appreciate, everything is complicated.
Yes.
I'll try to keep it simple, but the basic complication here is CRISPR actually isn't involved in the new vaccines, but the whole biotechnology enterprise and where it's been heading over the last decade or two is heavily implicated in both CRISPR and in the new vaccines and in every aspect of our coronavirus response. I mean, this is a good news, bad news story. The good news is, thanks to modern biotech, we are 5 years, 10 years ahead of where we would have been with a similar pandemic 20 or 30 years ago. Bad news is we still have 400,000 dead in the United States. So even with the new tools, they haven't stopped an enormous amount of suffering, both physical suffering, psychological suffering, and economic suffering. But I think they made it shorter. And the key here is to be able to play with nucleic acids. Now, nucleic acids is a broad term, but the two key ones, one of which everybody's heard of, DNA, deoxyribonucleic acid. And then the other one that fewer people have heard of is RNA, which is just ribonucleic acid. So DNA is missing an oxygen compared to RNA. That's why it's deoxyribonucleic acid. DNA is like it's the master recording. It's the master tape. It's the one that gets read off and turned into instructions, and I'd say turned into records and sent out to keep to turntables everywhere. But that's a dead metaphor. Although apparently vinyl's making a comeback.
It is. It is. I have a vinyl record player upstairs.
Yeah, so I got 1,000 vinyl records. I just don't have a turntable that works anymore. It's the master recording. It's really important, but it only functions through mRNA, through RNA, and RNA comes in lots of different flavors. The one that's involved in the vaccine is something called mRNA, or messenger RNA, which plays maybe the most crucial role in turning what's encoded in the DNA into proteins, which is what our body, for the most part, is made of and uses. So there are a lot of different RNAs. mRNA turns DNA's instructions into actual physical molecules called proteins. The vaccines traditionally have been made by taking virus vaccines. You can have vaccines against bacteria and other things, but sticking with the viruses. You take a virus and you either kill it, which is a little tricky because technically most of us don't think viruses are alive to begin with. They're this weird sort of thing at the edge between life and chemistry. But nobody has any problem talking about killing a vaccine, killing a virus, even if you don't think it's really alive. It's either a killed virus or a weakened virus, an attenuated virus. And the idea there is there's something about the virus that triggers your immune system to make antibodies and other things that attack it. Well, what this has done, well, I should say there are a whole bunch of vaccines under development, and they're running the gamut. There are 5 or 6 different ways to make vaccines, and every one of them is being tried with respect to COVID, including the good old-fashioned methods of dead or attenuated virus. But the mRNA method is to say, wait, we can figure out what part of the protein of the virus the immune system reacts to, and we'll pick a part that's always on that virus, and we think we'll get a strong reaction. And we can put this messenger RNA in the vaccine, put it in your body. Your body then turns that into the viral protein. That's just one of many viral proteins. It doesn't turn it into a virus. There's no virus there. There's just one piece of the virus, one protein from the virus. And your immune system goes, invader alert. Better make defenses against that. We're able to do that in part because we can manipulate mRNA, in part because thanks to gene therapy experiments and other things, we know how to sneak this RNA into your body in a way that the body will accept. And in part, and most importantly, because we can sequence the virus. We can discover what the A, T, C, G, D, T, 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 A, C, G, et cetera, of that virus is. It's like 19,000 letters, which is not very many. Think of that, turn that into words, that's about 2,500 words, so it's a long op-ed. It's a really short judicial opinion, and it's the intro to a brief.
Right.
That's the virus. And we know that now, which we wouldn't have known 30 years ago. And 20 years ago, it would have taken us a year, 2 years to figure out. And 10 years ago, it would have taken us 6 months to figure out. Now it was trivial. And it's so trivial that we can actually track each different version of the virus. Every time one of those letters gets misspelled, we can see. So it's that whole the whole development of the technology around nucleic acids, DNA and RNA, how to read them, how to make them, how to sneak them into the body, how to edit them. It's all part of our COVID response. The one part that really hasn't been part of the COVID response, ironically, is CRISPR.
Yeah.
Because CRISPR is about editing DNA, about changing DNA, and we haven't had to do that. But there is a wonderful, deep, ironic connection here. I know Jennifer Doudna fairly well. I've gotten to know her, and she well deserves the Nobel Prize, along with her colleague, Emmanuel Charpentier. And Doudna is also just a really thoughtful, nice, ethical, good person. I like her a lot.
Mm-hmm.
She did not invent CRISPR. And she agrees she did not invent CRISPR. Who invented CRISPR? Nameless bacteria 3 billion years ago or more. And why did they do it? To fight viruses. So CRISPR is a bacteria's way of fighting virus, just like the mRNA vaccines are our way, one of our ways of fighting virus. So CRISPR was originally an immune device, an immune system device to fight viruses. You might think viruses attack bacteria. Viruses attack everything. And they particularly love to attack bacteria. Bacteria have been around for 3 to 4 billion years, so they've had lots of time to come up with defense mechanisms. CRISPR is just one of them, but it's what Charpentier and Doudna did is figure out how humans could use it as a tool. Take this thing that bacteria had come up with and turn it into a tool. And it's a really useful tool.
Well, I watched the documentary that Dan Rather's production company put together, Human Nature, which is all about CRISPR.
Yeah, it's a fun piece. I like that.
It's really, really good for those who don't come from a science background like myself. It's a great introduction into exactly the kinds of things we're talking about. And in that documentary, you meet this yogurt culture.
Yeah.
Producer from, I forget, somewhere in the Midwest, and he had a role actually in helping to understand how to get to CRISPR as a tool to engage in the kind of gene editing we do today.
Right, so, because viruses were attacking the bacteria and the yeast they were using to make yogurt, and he was trying to figure it out. CRISPR is a wonderful story on many, many levels. My new book is really just about one aspect of it. Its use by He Jiankui, a rogue, I think, scientist in China, to CRISPR humans and make babies whose genes he had edited. It's a lot bigger than that. There are a lot of good books out, and there are a lot of books coming out about CRISPR more broadly. One of the great things about CRISPR is it shows that it really, yes, Doudna and Charpentier won the Nobel Prize. There were another 4 or 5 people who maybe could have been given that prize as well. Mm-hmm. But there were hundreds of people, thousands and tens of thousands in the long run, but even in the short run, hundreds of people whose work was crucial to the development of CRISPR, including the yogurt company, including this wonderful microbiologist in Spain named Francisco Mojica, who I was kind of hoping would win the Nobel. He was just studying it in these weird marine organisms that he saw. And his work was crucial to it. So science, every once in a while, like Einstein's theory of general relativity seems to have come out of nowhere. Everything else builds on everything else, just like you can't write a constitutional law decision today that isn't affected by the last 220 years of constitutional law.
Well, that's, that again brings us back to the vaccine, and then I want to broaden out to some of the other ways this technology is being used and could be used in the future. But what can you explain why you said that the development of these mRNA-based vaccines is part of the broader technological advances that CRISPR's been a big part of, but it isn't direct, but CRISPR itself as a tool for gene editing isn't directly involved in the vaccines.
Correct.
If we become increasingly concerned, and we should, about these variants that are now out there, these variants which are mutated strains of the COVID vaccine, of the COVID virus, would CRISPR be used to modify the vaccines that we have to make them effective against these new variants of COVID?
So you probably wouldn't use CRISPR to modify the vaccines because the stretch of RNA is small enough that you can just build it. So let me back up a second.
Okay.
Sort of, there are four big steps to the genetics or nucleic acid revolution in biotechnology. The first is being able to read code, read DNA, read RNA. Really, really important. Everything depends on that. The Human Genome Project arguably produced the first whole human genome, all billions and billions of letters in our genes. And each one of us and each of our human cells has, if each of the base pairs that make up the DNA was a character in a book, every one of your cells, each one of which is too small to see, has inside it War and Peace 1,000 times. The Lord of the Rings 1,500 times. So there's a lot of stuff there. And we're now, the first reading of that costs somewhere between $500 million and $3 billion, depending on which accounting methods you use. Accounting being, of course, the darkest of all the dark arts. Today, we can do that for a couple $100. And for the much smaller virus, we can do that for a tiny amount of money. That's what those tests that you get, that they stick something up your nose or back to make you gag, that's basically doing sequencing of that viral genome. So the ability to do that in quantity and cheaply, hugely important. That's reading. Then there's writing, building. We can build DNA. Now, our DNA comes on chromosomes that are millions of bases, hundreds of millions of letters long. That's hard to build. They've built the chromosomes for yeast. We're getting there, but to build 100, 200, 1,000, 10,000, 20,000 long stretches of DNA or RNA we can do. That's what the mRNA is doing. It's building this messenger RNA, and it's writing it very accurately, and then it's encasing it in fat, actually, goo, and sneaking it into the body. So you don't have to edit it because it's short enough that you can actually write it or rewrite it. Editing is what CRISPR does. And, you know, the best analogy really is, I think, well, CRISPR turns out to be the Swiss Army knife of tools. It does 20 different things, and each week somebody's discovering something new it does. But the basic thing it does is cut up DNA, and we use that to edit DNA. And you can think of the CRISPR as the find and delete function on your word processor. So my name is Greely, Greely. It's a problematic name because most Greelys are Greeley. Every once in a while, my law school, where I've worked for 36 years, misspells my name. So if I'm in something and somebody's misspelled it and I'm editing it, I can just tell Word find Greely and delete. That's basically what CRISPR does. It says find this stretch of bases of DNA letters and cut them out. Then what you can also do with word processing is say replace, find Greely, cut it out and replace it with Greely. CRISPR can do that too. So it's the editor. And then the fourth step is to know what the hell it all means. And ironically, that's the one we've been doing longest, but we know least about. We know a lot about some stretches of DNA, but most of DNA is still how it works, how it functions, what it does, still pretty mysterious. So, yeah.
Well, that's kind of
read, write, edit. CRISPR's the edit part. The mRNA is really the write, but it's based on all the reading.
Well, that seems, as I understood it, that was really the source of the big questions that this documentary, Human Nature, asks, is that when you edit, when you delete and replace, to use your analogy, you don't always, and maybe hardly ever, know what the impact of that deleting and replacing is going to have on that organism.
Yeah.
The big problem.
I teach 1Ls from time to time, and one of the things I always try to tell them, well, first thing I tell them, they always ask, can they sue? To which my answer is always, they can always sue. That should never be your question.
A ham sandwich can file a lawsuit, we always say.
Right. It may not have standing, but if it's got a lawyer, it can file a lawsuit,
right,
or it's smart enough to go pro se. But the other thing I tell them is the best 2 words to start any answer to a legal question are always, it depends.
Mm-hmm.
So some genetic stuff we understand really, really well. We understand what it causes. We understand what making this modification will do. So let's say the genetic variation that causes sickle cell disease. It's a 1-letter change.
Mm.
1 letter. I think it's an A that becomes a G. I don't quote me on that.
Mm-hmm.
But it's a 1-letter change. And we know not only that 1-letter change will cause sickle cell disease, but we know why it will cause sickle cell disease, because it changes one of the pieces of the protein that the gene makes in a way that changes the shape of the protein. And it's a protein that's a globin protein and hemoglobin, which is in blood, which causes your blood to mess up. Yeah. We understand everything about that. Most of the genome, we don't know anything about. We don't know whether it does anything at all. And the things that we know do something often do a bunch of different things. So the gene that was involved in the CRISPR babies in China is a gene that almost all vertebrates, so fish, actually I guess fish don't have it, birds, reptiles, amphibians, and mammals all have this gene in slightly different forms, but it's all basically the same. It's all pizza, some of it's deep dish, some of it's thin crust, but it's all pizza. We know that it gets involved in the immune system. We know humans can live without it. We don't know how healthily they live without it, but we know that HIV needs it in order to infect a part of the immune system called the T cell. So we understand that. And so what He did was knock out that gene in these babies in the hopes that they wouldn't be able to get HIV infected. But we don't know what else that did.
Mm-hmm.
And there's some evidence that it makes people more susceptible to bad cases of influenza or bad cases of West Nile virus. And the fact that every mammal has it kind of makes you think it's probably important, but we don't know. So our level of knowledge ranges from complete detailed understanding to complete ignorance. And most of the time it's somewhere in between.
Well, let's stick with the sickle cell analogy or example, cell example, because that is a good one, because what it shows is that this technology can and is close to really solving the problem of sickle cell disease. You have sickle cell anemia. That's a terrible disease. It's terrible for that person. It's terrible for that family. It's terrible for society, you could say. It's, in so many ways. So on the one hand, you can say, this is an incredible medical, scientific, technological breakthrough to be able to read the code, to write the code, to delete and edit that code, to cure a disease as terrible as sickle cell anemia. And that ought to, and I think is providing tremendous motivation to many companies and many scientists around the world to use CRISPR.
And many people with the disease and their families and disease organizations. I mean, yes, everybody's in favor of curing disease.
But we are going to get into how this then relates to questions of how we think about human creation and maybe what Cat Stevens called recreation or recreation, betraying my 70s origins musically. What are so from an ethics, medical bioethics standpoint, then obviously we're talking about this. It's not just a black and white, clear cut. It's a definite good and only a good to think about eliminating sickle cell anemia. So what are the parts of it, at least potentially or theoretically, that are not so good?
Yeah. So again, those two words, it depends.
Right.
Even for good medical treatments of awful diseases that don't have other treatments, and sickle cell pretty much falls into that. And by the way, we have not yet approved gene therapy for sickle cell, but it's getting close. My colleague, Matt Porteous, who I think is in that movie, is doing a lot of research. He's got a couple of patients who've been basically well for a couple of years now. And we will get there. And FDA has approved a couple of gene therapies, typically for really, really rare diseases, but really rare and bad diseases. Everybody's in favor of that. But even that has issues like if the company is charging $2 million a treatment, does everybody get access? Who gets access? Who ends up footing the bill for it? If your law firm has a health plan for your employees and one of your employees needs this, does that mean you have canceling the health plan for everybody because you can't afford it anymore? Mm-hmm. Are there racial or other discriminatory issues in which diseases get taken on first? And although sickle cell is happily getting a lot of attention now, I think one could argue quite fairly that for many years it didn't get much attention because in the United States, it's largely, though not entirely, an African-American issue disease. Mm-hmm. So there are always ethics issues. Even with a really good thing, there are always ethics issues. But there aren't deep ethics issues when it comes to using genetics to treat disease. Nobody that I know of says, well, maybe some anti-vaxxer types would say this, but basically no one says, no, it's deeply unethical for us to try to cure disease using gene therapy. Christian scientists might argue with it, right? But that part is fine.
Well, you probably have a small contingent of Malthusians who worry about overpopulation of the planet.
Right. Although, yeah, that's a different edition of this podcast.
Right.
But there are two ways in which it gets trickier. With sickle cell, you're changing the genes, and it's one gene that operates in one kind of cell whose job it is to make red blood cells. And you're doing it in a living person. And basically with that, you're taking bone marrow out, getting the stem cells in the bone marrow that form blood, editing those to turn that A into a T. And in fact, it gets trickier than that because there are other ways people are trying to do it using CRISPR to turn back on fetal hemoglobin rather than adult hemoglobin. It's like the tax law. Everything is more complicated than you think it's going to be. But ultimately, you get through it, or you don't. The two problems are, first, if you do it in a living person, it dies with that person. It's what we call somatic cell gene therapy, where somatic is just a tricky word, a fancy word for body. What's not a somatic cell? A germ cell. What's a germ cell? An egg or a sperm. Why are they different? Because they can, in theory, live forever. They can get passed on to the next generation. Mm-hmm. So if you're just changing blood-forming stem cells, it dies with the patient. If you're changing the cells in the early embryo, that gets into every cell in that person's body: heart cells, lung cells, blood-forming stem cells, eggs, or sperm. And that can pass on from generation to generation.
Right.
And so
to take that patient who's got sickle cell anemia, for instance,
right,
the technology is at a point where it's conceivable and we're close to being able to cure that person of sickle cell disease, but not yet to prevent that person, even after cured,
right,
with passing on that genetic code for sickle cell disease to.
That's exactly right.
Okay.
So it may be that person, so sickle cell is what we call an autosomal recessive disease. The only thing you need to remember about that is you need to get a bad copy of the gene from each parent. 2 bad copies makes you sick. 1 bad copy, not a big deal. 1 in 10 of European Americans carries a bad copy of the gene for cystic fibrosis. One bad copy, no problem. One bad copy marries somebody with two good copies, no problem. Person with one bad copy marries somebody with one bad copy, on average, a quarter of their kids will get two bad copies, problem. And even if you did gene therapy to fix cystic fibrosis in the living person, doesn't affect their eggs and sperm, doesn't pass on. And so if they have a kid with that, that kid will have to get the gene therapy too. But the good news is that sometime in the future, and the gene therapy will only get cheaper and easier and more effective, et cetera. So that's right. Lots of people think we should draw a line between gene changes in humans that affect only the person who's getting it and gene changes that affect future generations. Now, even that line is a little tricky, because if I cure your disease, I'm not just affecting you, I'm affecting everybody who cares about you. Everybody who loves you, likes you, or even people who hate you, arguably, are affected by the fact that you've been cured from this disease, as are the doctors who treat sickle cell disease, as are the stockholders of the companies that sell the cures. And on the other side, just because you could pass it down to your kid, maybe you won't have kids. Maybe your kids will get the unedited version of the gene. Maybe your kids won't have kids. It's not that changing somebody's eggs and sperm necessarily means the entire species is going to change. So, but that is sort of the extreme idea. On the one hand, you affect only one living person. On the other hand, you potentially affect everybody. That's one of the big divides. The other big divide is what you're using it for. Almost nobody has a problem with curing diseases. Lots of people have problems with what's called enhancement. So we want to make you taller, we want to make you faster, we want to make you smarter, whatever that means. We want to make you better at math, we want to give you perfect pitch, which turns out to be partially but not totally genetic. We want to give you curly hair instead of straight hair. We're going to give you blue eyes instead of brown eyes. We want to do something really good for you and give you the gene that causes your hair to turn perfectly white at an early age, which is a really useful enhancing trait. My family thinks so, anyway. And the silverback gorillas rule the gorilla bands, and I think it's because of the hair. So it's the enhancement, and at the far end, it's the X-Men, right? It's the mutant superheroes, et cetera.
Who don't feel pain. In other words, you could edit so that you... So
I'm so glad you mentioned that. It's a bit of a digression, but not much. There are people who don't feel pain. It's a terrible disease that's genetically caused that people are working on fixing, because if you don't feel pain, you usually don't live that long because you don't realize that you shouldn't put your hand in the fire. Pain is actually, to some extent, it's a useful thing. So this is a nice example of, bad things. The line between bad and good is sometimes tricky. So it's the enhancements that get people most nervous. And the enhancements in a living person get people nervous. And so the ICOC is really worried about the IOCC, International Olympic, the Olympics. IOOC is really worried about gene doping. Not clear anybody's done it or could do it, but if you gave somebody an extra copy of a gene that makes hemoglobin, it's like giving them the drug erythropoietin or giving them blood transfusions, and in theory improves their endurance. That's enhancement, that's somatic enhancement. Their kids don't get it, but they get it. And then people also, and the thing that worries people most is germline enhancement, where it passes down from generation to generation. And here's where you get all the dystopian novels and, the genetic elite versus the genetic lumpenproletariat. It's kind of a version of Gattaca, although Gattaca wasn't about editing as much as it was about selecting. It's kind of going all the way back to H.G. Wells and the Time Machine, the species turning into two different species, the Morlocks and the Eloi. And so serious people worry about CRISPR leading to a speciation event where humans turn into different species.
So that brings us back to China then, and your books, but why did the scientists in China get in trouble? I mean, what was the line? Because if there was a place where they were going to cross the line, ahead of the rest of the world. I would think it would be in China in today's, today's culture and politics. So why did that team get in trouble?
So it's really interesting. And I do want to say a word in defense of China. They actually follow the same general rules of medical ethics and bioethics as pretty much everybody else. A few exceptions. They don't worry as much about embryos as we do. They don't have a right to life movement the way we do. Their animal treatment is they're not quite as concerned about how animals are treated, but by and large, they have the rules. They don't implement them very well. I don't think, I can't obviously guarantee it, but I don't think He Jiankui could have done in the US what he did in China, because they're weak on the ground in implementation. There are 1.3 billion of them, and they've only really had laws for about 30 years. Before that, it's whatever the party did, and the statutes didn't matter. So they don't have the sort of depth of structure organizationally that we have. But what's also interesting, the first news story out of China after this broke had headlines lauding this wonderful breakthrough in Chinese science, how this was such a great thing. That same day, 122 Chinese academics posted an open letter on WeChat, which is their version of Facebook, saying this is a terrible thing, and it has shamed our country in the eyes of the world. It has made us look, evil and weak. And so I think it was very clever of them to play this sort of nationalism card there.
Right.
And then within a few hours, the news stories coming out of Beijing where terrible thing happens, investigations going on. I think the Chinese government might have gone the other direction on this. But instead, they joined the almost universal attack on what He did. And the attack, there's several different levels to it. The people who think any germline modification in humans is a bad thing, they all dislike it because it's germline modification in humans. It's a modification that can pass from generation to generation. These two little girls could, in theory, pass that gene on, and in 10,000 years, every human in the world would have their version of the gene. Won't happen, but theoretically it could. So the strong principled opponents hate it. But then there are a bunch of people who don't necessarily have a strong view about the principle, but this use of it, and I'm in this camp, this was criminally reckless. Nobody had ever done this in humans before. The first time you try anything in humans, you need to pray to all the gods you believe in and throw salt over your left shoulder and cross your fingers, and because nobody knows how it's going to turn out until you try it. And that means you need to be as close to confident. You need to have done a lot of animal work. You need to have done a lot of in vitro work. And you need to do it only in situations where there's a really good potential benefit. This was incredibly risky because it's something that's never been done in humans, barely been done in non-human primates. The particular thing he was doing had never been done in non-human primates. So the risks of the health of the babies. I'm not worried about the future of humanity. I'm worried about these two babies. The risks to them were extremely high and the benefits were close to zero. He was trying to limit their ability to be infected with HIV. He thought he was going to eliminate it. With one of them, he didn't eliminate it. He may have reduced it. With the other one, he certainly didn't eliminate it. He may have reduced it a lot. We don't know because the Chinese government has not let anybody outside examine these babies. We don't know anything about their health since the time 6 weeks after they were born that He made his announcement about them. And that's over 2 years ago. So very risky to the babies, very little benefit because A, it's not going to prevent them from getting HIV. B, they're probably not going to get HIV. C, if they are, it'll be 20 years from now when it's likely to be even better treated than it is today. There's no real benefit to them. There's huge cost, huge potential risk. And that made this, I think, and I think almost everybody agrees with me, really criminally might be argued, but unethically reckless.
Mm-hmm.
There's also the case that they appear to have forged the permission slip from the Human Subjects Ethics Committee. That's a problem.
Definitely.
Chinese law does not allow people who are HIV positive to use IVF. I think that's a bad law, but that's the law. So they apparently hired guys who were HIV negative to pretend they were the parents who were providing the sperm. That's against the law. That's a problem. To the extent anything in international science has a consensus, this is too risky to try now. Some people think never, some people think maybe in the future. Everybody thought, with one notable exception at Harvard, everybody else thought not now. He ignored that. And everybody said, if you are going to try this, make sure you're public about it. Everybody knows about it. There's lots of discussion. And he did it in deep secrecy. So what he did was unforgivable. Now, was it illegal? So forging the IRB certificate, it's going to be illegal.
That's the review board that would oversee the human research ethics.
Yes. Yeah. Thank you for translating. I try to be good at it, but every once in a while I slip.
Institutional review board.
It's like when we're talking to non-lawyers and we say, oh, strict scrutiny or racist loquitur or so on. You got to remember your audience and vocabulary. So that's illegal, but they actually, all we knew about his trial was the announcement on December 31st of 2019, a year later, that he had been tried, found guilty. It was not an open trial because of worries about the privacy of the babies, but certain, comrades were invited to the trial. All we have of it is two columns and a press release from the Chinese official government press agency saying they confessed to their crimes. We've never seen the indictment or the civil law. China has a civil law system, not the US adversarial system, but we've never seen the charging document. We've never seen a verdict. There was not a jury, of course. Apparently, according to the press release, they were convicted of unauthorized practice of medicine and sentenced to 3 years in prison. But of course, we don't know where he is. Nobody's heard from him. Yeah. Maybe they've decided to denominate his lab a prison and he's still working in the lab. We don't know. Right. Because, frankly, I don't think the Chinese government is going to tell us anytime soon, which is one of the really scary things about writing this book. Now, how do you write a book about something where there's basically no information? Nobody even really knows whether these babies exist. No outsider, nobody outside China knows whether they even exist. Right. So that's a little frustrating. Plus, in the delay between finishing the book and when it actually drops, there could be a news story still tomorrow that comes out and makes my whole book meaningless.
Well, let's get into
not for the faint of heart, this book writing stuff.
Let's get into that and the book, by the way, is due out this month in just a couple of weeks. And as we always do, we'll put a link to that in the description for this episode. And I know I'm looking forward to getting it and reading it, no matter what news stories come out in the next week. But you defined a couple of different camps when looking at what went on in China. Those who you said had a principled opposition to any germ cell editing, that is any kind of gene altering that could be passed down, as was the goal of the experiments that were done in China.
Yeah.
Because those people would say, you don't know what the unintended results may be. Countless iterations of that played out through generations, out.
You're making a decision potentially for all of humanity, and you're not asking humanity's permission.
And then you said there's another group that looks at what happened there and says, under the right circumstances, and for the right purpose, I can see that the benefit outweigh the benefit may outweigh the risk. I remember reading a short story when I was a kid. I believe it was by Ray Bradbury, that now we refer to as the butterfly effect. A guy goes back in a time machine for a little holiday, and goes back to prehistoric times. And the tour guides who take him back on this trip say, whatever you do at all costs, you must remain on the path. Do not step off the path. And the guy, being reckless, accidentally steps off the path and accidentally steps on a butterfly and thinks, well, it's just a butterfly. You know what? I know I wasn't supposed to step off the path, but what could possibly be the outcome of that? He goes back in the time machine to go home, however, hundreds of thousands of years later, and the entire civilization is completely upside down and different.
Yep.
All because he's so. I guess that's close to what you were saying, the people that think, well, we don't know what the unintended outcome of that kind of gene altering could have on our species or on our planet, really, because humans are so interconnected with our environment, with our environment, small and large. But also, once you cross that line from just treating the cells of a body to treating the cells of a future body,
right?
And haven't you given up control, or because then the genie's out of the bottle. So it's the unintended outcomes that might result that we can't imagine or foresee or predict in any one application of the technology in this way, but also
what other applications is opening the door
maybe
for?
Have you lost control of designer babies? Have you lost control of a dictator creating an army of soldiers who don't feel pain? How do you put that back in the
So I think that's not a crazy argument. I don't accept it myself, and I've got a chapter in the book talking about, is germline editing inherently unethical?
Yeah.
One of the problems with it is, of course, everything you do has potentially unpredictable consequences, and that includes not doing something.
Mm-hmm.
The Europeans love the precautionary principle. Don't do anything unless you're absolutely confident that could have irrevocable consequences. But the problem with that is not doing something is an action.
Right, that's true.
So part of it depends on what you think is technologically possible. Many of the people who are worried about it would say, well, the medical stuff, that would be fine, but people will use it for enhancement. So that's the slippery slope issue of different uses you were getting at. And that, one reaction is, well, let's regulate enhancement. And some people will say, well, I don't trust the world to regulate enhancement. But of course, if you don't trust the world to successfully regulate enhancement, do you trust the world to successfully keep anybody anywhere in the world from doing the germline stuff to begin with?
Mm-hmm.
So it's complicated. But it does have potential long-range effects if you think we can't control how we use things. Well, anybody who thinks they can control anything is kind of a fool.
Right. So we may be already, the genie may already be out of the bottle.
But the question is, how likely are we to be able to control something? And what can we do to try to control it? And I think that I feel fairly confident that we could control uses of this. You still have to worry about the immediate consequences. Does changing this gene turn out that if you change this gene when somebody turns 65, they drop dead? Because we've never seen this change before, and maybe that happens, which is always a risk. It could be that every IVF baby will drop dead at the age of 43, because Louise Brown, the oldest, is only 42. Mm-hmm. We don't know. Yeah. So I use that as, I think that's a really good argument for continued monitoring, assessment. And our society, it's like, I'm at a university. It's relatively easy. It's hard, but it's relatively easy to raise money to build a building. It's impossible to raise money to endow the janitors for the building or the maintenance for the building. Mm-hmm. And we're good at doing things, but we're not so good at making sure we follow them up and monitor them and adjust them as things go on. And I think that's a big story. But that is the biggest concern, I think. Well, that's one big concern of the principled objectors. Where will this all lead? Can we stop on the slippery slope? in general, I'm not keen on slippery slope arguments because slippery slopes are everywhere. When a student brings it up, I ask, so have you ever driven on Highway 280 here in the Bay Area on a nice day when the traffic's clean? If you go 65, which is the law, you get killed. You get rear-ended. You're breaking the law. Is that on the slippery slope to becoming a mass murderer? Well, it's breaking the law. You're on the slippery slope. Figuring out how to hold our place, find and hold our place on the slippery slope is kind of what we do. There's another principalist objection, which is kind of the God or nature objection. The religious version is you're playing God. The naturalist version is you're playing Darwin or you're playing nature.
Yeah.
And they both are, don't intervene, don't change. And I think part of it is this sense of the human germline genome is like the Ark of the Covenant. You know, it's really powerful, it's really sacred. And the one thing we know is we can't let the Nazis get it, right? Indiana Jones.
Right.
But part of what my chapter goes into is there's no such thing as the human germline genome. There's 7.5 billion human germline genomes. Yours is different from mine. My kids is different from mine. Identical twins have even subtly different genomes because of mutations that happened after they split off as embryos. And those mutations, the germline genome changes every generation because of mutations from the time you were conceived to the time you made the sperm that made some of your kids. So I don't have exactly the same gene sequences that my parents got and my kids don't have the same ones that I got. And humans affect how that goes. We have different genetic variations because we invented agriculture. And that's literally true. We have more copies of a gene that helps digest starch than our ancestors had because agriculture is an advantage to digest starch. Another way to see this is populations that did a lot of dairying. They tend to be adult lactose tolerant. Most of the world, once you become an adult, you don't digest lactose well, you can't drink milk, but there was a mutation and the people, and different mutations, it arose separately in different parts of the world, they got into dairy. So for them, if you're in a culture that's dairy, it's an advantage to have the version with dairy. And so the germline, the overall incidence of the pro-lactose gene increases over time. Medicine, type 1 juvenile onset diabetes used to be deadly to people, and it was called juvenile onset because it hit between usually 5 and 15. There's a genetic connection to it. It's not a complete 100% genetic connection, but it's a moderately strong one. Those people didn't used to be able to pass it on to their kids because they didn't live long enough to have kids. Then these interfering Canadians, these blasted Canadians, 100 years ago discovered that they could take insulin and keep those people alive. Interfering with nature or God. And so my cousin Mike, who's exactly my age, has something like 14 grandkids. He was diagnosed with type 1 diabetes when he was 10. But for those blasted Canadians, he would have died before he could have had kids. His risk gene for type 1 diabetes has been pushed more broadly in the population than it would have been except for medicine. So we are changing the overall human germline constantly, if somewhat indirectly. And this gets into this weirdness that humans have, we often have. We're willing to accept sort of random stuff outside our control we don't know about, we didn't control it. But if humans try to do it, then we get much more nervous about it, even though the humans at least can be trying to do it for good reason. Maybe we won't do it right, but nature is out there and it doesn't care whether it's a good reason or not. COVID is almost certainly, I'm not a believer in the conspiracy theory of SARS-CoV-2. I think it was a natural variation in one of the gazillion forms of coronavirus, but nature is a terrorist. At least humans are going to usually, except for the bioterrorists among us, try to use this for good ends. So I think reasonable people can hold to the it's a bad thing, period position. I don't think they really should, but lots of people believe things that I don't think they should, and they're still reasonable, intelligent et cetera, people. I think the more interesting chapter in the book in some ways, I have this personality defect. I am drawn to a middle position in any argument where I get shot at by both sides. Mm. It has the advantage that you don't have to ask, which way should I look, because the incoming's coming from every direction. I also don't think that the germline gene therapy is going to be gene editing is going to be very important in humans, because I don't think there's much use for it. Almost everything that you could do, that you'd want to do, you can do through a technique that's been around for over 30 years called pre-implantation genetic diagnosis, where you have to use IVF. That's a problem, in vitro fertilization, but it's not a huge problem. 200,000 Americans use it every year. And then you take the little embryo and you test genes from a few of its cells, and you decide, this embryo will get sickle cell anemia, this embryo won't. Let's transfer the embryo that won't. Mm-hmm. Rather than edit the embryo, which adds another technological step and more risks and more money, just pick the good embryos, the healthy embryos versus the sick ones. The oldest kid born this way is over 30 years old. It's safe and effective and well known. My earlier book, with the more titillating title, The End of Sex and the Future of Human Reproduction, is really all about that. Mm-hmm. I think between pre-implantation genetic diagnosis or fixing the disease, the other thing you could do is, rather than do gene editing to avoid the disease, wait till the baby's born and then use gene therapy to fix it. Mm-hmm. If our gene therapy gets good enough and it's headed in that direction. So I don't think there are going to be a lot of needs for germline editing. Enhancement probably would be one, because any kind of enhancement probably would involve multiple genes. And the more genes you've got, the better editing looks as compared to selection. But the issue there is we don't know squat about what genetic variations enhance and don't enhance. We know literally hundreds of genes that are strongly involved in intelligence. If the gene is broken, you have very low intelligence. Mm-hmm. We know almost nothing about genetic variations that increase intelligence. The most optimistic researchers in that field think that maybe they understand genes that can explain 1 or 2 IQ points. And those are the optimists.
But physical characteristics, though.
Yeah.
Those would be a lot easier to manipulate.
Yeah, kind of, although it's trickier. Even that's trickier than you might think. Eye color turns out to be 10 or 20 different genes. But there's one powerful one that's dark eyes, light eyes. And that's pretty clear. Height turns out to be hundreds of genes. And even knowing the hundreds of genes, the best way to predict a baby's height is to know the adult heights of its parents and whether the baby is male or female. It's much better than looking at all the genes, ironically. So we will get better at things. And I think the cosmetic traits are likely to be better. Anything behavioral is going to be really tricky, because our brains are just, I also do law and neuroscience, in part because genetics, we might figure out in my lifetime. Neuroscience, we're not going to figure out in my lifetime. Brains are like 10 times or 100 times more complicated than genes. And so any enhancement involving behavior is going to be really complex. I say that, although we do know about ways to enhance intelligence. You give kids a good diet when they're young, you pay attention to them, you keep them healthy, you read to them. Those are all ways to enhance intelligence. We just don't know anything about the genetics of it. I don't know whether we ever will.
That's so old school, Professor.
Yeah, I know.
Take care of your kids.
It's not sexy.
Right.
It's just you mentioned earlier, you're a parent, right?
Yeah.
I mean, that's part of our job. That's our biggest duty, is help turn these ignorant barbarians into good people.
Right.
And we don't do it with genes.
Well, so much to think about, and I know I've already kept you longer than I had said I would, but I continue to learn and think about this, and I've been doing a lot of learning and a lot of thinking about this as we've been talking. So I want to thank you for that, Professor Hank Greely from Stanford, and also again highlight the book that you have coming out called CRISPR People, Science and Ethics of Editing Humans. Another, maybe not quite as sexy as The End of Sex, but a very provocative title. And I think based on the conversation and the way you're able to talk about and explain and actually also inject some humor into the conversation as well and broaden our perspectives. I know I'm going to look forward to reading the book.
So Aaron, can I throw one more thing in?
Yeah.
So what I think is most important with CRISPR, I haven't written significantly about yet. That's the next book where I've got all of 3 words written, the title, Playing with Life. I think it's really going to be a big deal as we intentionally change the genes of non-humans, whether it is cattle to make them better as cattle, whether it's to make them environmental, belch less methane or be tastier, whether it's mosquitoes to keep them from passing along malaria, which people are working on, whether it's algae to make them sequester more carbon from the atmosphere. We're going to do that, and we're going to do that a lot faster than we're going to deal with humans, because if something doesn't work and 50% of the mosquito babies die, nobody's going to get very upset.
Right.
And we're willing to take much bigger risks, and that worries me. So I actually think, at least in our lifetimes, the non-human uses of CRISPR are going to be more important. And because we're such an egocentric species, we pay enormous attention to ourselves and not nearly enough attention to the rest of the biosphere. So given my past record, I'd say, 5 to 8 years, long gestation period, but I hope sometime that we can come on and we can talk about my new book, Playing with Life.
That sounds great. I will look forward to that. Thank you so much again, Professor Hank Greely from Stanford. It's such a pleasure to speak with you and get to meet you today. Thank you again.
I enjoyed it greatly. Thanks for giving me the chance.